I work at the Institute of Metabolic Science in Cambridge, and in it we essentially try to ask two sets of questions. One, why are some people fat and some people thin? And the other is, when you get fat, why do you get sick? And we try to use that information to better inform prevention and treatment of important diseases. But of course, obesity and type 2 diabetes, which is largely the most important metabolic complication, these diseases have increased dramatically over the last 30, 40 years. And this has got nothing whatsoever to do with our genes, this recent increase. So it's perfectly reasonable to ask an investigator like me, who uses a lot of genetics to try and explain these disorders, what on earth am I up to using genetics to try and examine important questions related to these disorders? It's a reasonable question to ask any scientist, is why are they bothering? And broadly in science, I think there's two reasons to bother. The first reason to bother is that what you might be doing might be useful. Now, how might genetics be useful? Genetics might be useful in giving people diagnoses, whereas previously they only had broad terminologies for the disorder. People don't like that. People like to know what's wrong with them. And by giving specific molecular diagnoses, that can improve patient well-being. People like to know what's going to happen to them in the future, and molecularly characterizing diseases can give you a much better idea of what's going to happen and what's the broad spectrum of issues that are going to face someone in the future. We'd love to be able, and are increasingly be able, to use genetics to target therapy to specific molecular mechanisms. And finally, a long-standing aspiration of genetics is to be able to take healthy people and use genetic information to predict which ones are at risk of particular diseases or not, and therefore influence their behavior. But of course, scientists don't just do science because it's useful. Scientists do science because it's interesting. And of course, genetics can be very interesting. It can actually help teach us how we evolved from primitive organisms to the organisms we are today. It can teach us how we get from a single cell into a complex, developed multicellular organism. And importantly, it can teach us a lot about human physiology, about where are the particular control points and homeostatic pathways that control our function as an adult organism. Now, when you think a little bit more deeply about that, can we do anything with human physiology? Well, we do it all the time, those of us who are practitioners. If you treat hypercholesterolemia or hypertension or hypercalcemia, you're constantly tweaking those physiological control points that have been identified through studies of physiology, pharmacology, but increasingly genetics. And genetics is proving a very good way of discovering many of these control points in our physiological pathways. And by doing so, the interesting can, if you like, become the useful, and we can use that information to target and study and develop therapies targeted to particular important control points. But of course, if your idea is that you want to use genetics to understand physiology, the first thing you might think is human beings are a terrible species in which to do that. I mean, quite rightly, we cannot control human breeding or genotyping. We have unconscionably long generation times. We can't follow generation after generation in the laboratory. And we can't manipulate the human genome in a tissue-specific or a temporally controlled fashion. So if I was a pure scientist, I'd be thinking, what a terrible species to start working on. But there are some reasons why studying humans might have some advantages, other than the fact that we're most invested in them. Some of our physiology is not paralleled by other organisms. As observers of the human status, doctors communicate with each other about rare cases. Unlike other species that we share the planet with, we have actually a unique ability to access rare variation across the planet through organizations and conferences like this. We've got very good physiological tools for studying humans, and indeed, we can only really study things like emotion and higher cognitive purposes to any really meaningful extent in species that we can speak to. And finally, there's some advantages in us being hugely outbred, in that if we only find even a small number of individuals with a severe phenotype and a severe mutation, because we're all got so otherwise outbred, it's very likely that those mutations are causative. Whereas if you study a single mouse knockout, even study 1,000 offspring of that knockout, you're still studying 1,000 identical twins in identical genetic background, and what may be precise in that background may not be accurate across all species, all strains in that species. So there are some reasons, I think, to study and exhort the study of humans. So broadly, if you're going to study humans by genetics, what approaches do we have? We have, I think, sort of parody. We have the butterfly net, a butterfly collector. We can go and study rare, extreme variation, extremely uncommon, but sometimes very illuminating and beautiful. Or we can be the multinational industrial sweatshop operator, studying hundreds of thousands of people around the world with small genetic variation but occurring commonly and having modest impact on large numbers of people. And both of these approaches have been very illuminating over the last 10, 15 years. And I'll mostly talk about the first, but touch on the second. Now, my talk is brief today. It's only 30, 35 minutes. And I thought that I'd tell you what I'm going to tell you before I tell you it, and then tell you it at the end. And essentially, what we're trying to use genetics for is try and put together the pieces about what controls these homeostatic control points and where the important information is about how to avoid type 2 diabetes. So how you might avoid type 2 diabetes is maintain energy balance, and that probably is something that your brain's very important for, i.e. controlling the energy in versus energy out on a long-term basis, and therefore not becoming fat. However, if you can't avoid getting into positive energy balance, the next best thing to do is safely store that energy as adipose tissue, because that's the professional tissue that was designed to store our excess energy. If that starts to fail, what's important in the light of that is to keep as insulin sensitive as possible the tissues which are most important for insulin action, which is skeletal muscle and liver. And also important, at least in terms of avoiding diabetes, if not some of the other complications of the metabolic syndrome, is to have healthy pancreatic beta cells that can continue to fight and produce insulin in the face of insulin resistance for long periods of time. And what I'll return to in the end is, I think, with a perspective of human genetics, bring this diagram back and see if we've had any alterations in the prisms through which we visit that diagram. My entry into the field of obesity genetics was a rather abrupt one, precipitated by a single patient. Before that, I think I shared with most clinicians a view that obesity was the cosmetically unfortunate end result of a series of adverse life choices, and that actually it was a bit of a shock to me to discover that there might be molecular controls of body weight. And the late 1990s was my road to Damascus conversion through a single patient in which we discovered that prohormone convertase 1 was the source of her lifelong obesity, and rapidly followed by that the human leptin deficiency had the same phenotype as the recently discovered mouse leptin deficiency, resulting in severe obesity. And therefore, in the late 90s, we had the first human beings whose obesity was explicable on a molecular basis. That precipitated a very dramatic era of discovery for me and my wonderful young colleagues who then joined me, and I particularly mentioned Sadaf Faruqi, Giles Yeo, and Tony Coll around that time. And we and others around the world went on an exciting journey discovering multiple genetic defects that all resulted in severe obesity in children. And without going through each of these in detail, I'll simply say three things. First, we didn't know this at the beginning, but all of the genes that we found tended to affect brain function and be highly expressed in brain. Secondly, what we did was take an agnostic attitude as to how they were causing obesity, and we brought the children into our clinical research facility. And with the great talent of Sadaf Faruqi working with these challenging children, we studied their energy intake and energy expenditure, and by and large showed that all of the defects we showed, in every case, the children had defective appetite. They were unable to perceive fullness and satiety in the same way as their wild-type siblings were, and therefore they overate. And therefore their obesity, although genetic, was behavioral, something that is very difficult to get through to the lay public and often difficult to get to doctors, that something can be purely caused by a genetic defect and yet influence very profoundly a human behavior. The third thing to say is that some of these diseases were not rare. Melanocortin-4 receptor deficiency affects about 1 in 1,000 people in most societies that we measured it in. So it's commoner than cystic fibrosis or Duchenne muscular dystrophy as a genetic condition. We, of course, had the great good fortune to be able to use leptin to treat children with leptin deficiency, and although this is rare, this proved the point that we could actually find a molecular cause of obesity and do something about it. And although there's only 30 or so kids worldwide benefiting from leptin treatment, it is a dramatic and life-saving therapy. It's also taught us a huge amount about the physiology of leptin in the human, both in terms of its effects on the brain and its effects on the periphery. In recent years, Sadaf Faruqi now leads our program of research in severe childhood obesity, and her Goose cohort is now more than 6,000 children in size, and gene discovery continues. About 1% of our very obese kids have mutations in the transcription factor SIM1. A rare few have got an accessory protein for the melanocortin-4 receptor, MRAP2 mutations. SH2B1 is particularly interesting in that in this condition, children who have mutations in SH2B1 have a broader range of abnormal behaviors, including sometimes aggression, showing that the CNS phenotype, not necessarily restricted to that of obesity. And KSR2, a condition in which hyperphagia in childhood is associated with lower energy expenditure measured in adults, one of the first conditions where we consistently find that. And new genes are coming through. We've just completed 1,000 exome sequence in collaboration with Inez Barroso at the Sanger Institute, and there are more genes to find, and there are more things to do. So we have not fully trawled the causative patterns of obesity in childhood at this stage. Is it relevant to common obesity? Well, what we're showing here is data from my colleague Ruth Lewis, who was in the Institute of Metabolic Sciences, now at Mount Sinai in New York, and Ruth has put together on this a list of gene SNPs that associate with common obesity. And the only point I really want to make here is that many of those genes overlap with those genes that we found in severe obesity, i.e. bad mutations causing severe obesity and many common variants nearby contributing to the range of BMI seen in this room. Even greater sized data sets have now been analyzed in a paper that Ruth was co-leader on earlier this year. This is just showing the tissues in which gene SNPs associated with obesity are most profoundly affecting gene expression. And what's totally dramatic about this is, you can see, when it comes to body mass index, really the nervous system is utterly dominant in terms of where these genetic variants are affecting the expression of their nearby genes. It really confirms what we showed in severe monogenic obesity, that the genetic architecture of BMI that varies of people across this room are essentially variations in the expression of genes in your brain, and where your brain wants you to sit on the body mass fat storage spectrum that we have. So I'm going to conclude the part on genetics of human obesity by asking, first of all, going back to the useful and interesting paradigm, has it been useful? Well, in severe childhood obesity, we have got more accurate diagnoses and prognoses. We have got one rare disease that we can treat, and already we're moving towards working with companies and working with others on the basis of the logic behind some of the molecular defects that we found doing targeted therapeutic trials based on disease mechanism in the other diseases. In common adult obesity, it's not yet a clinically useful risk predictor, nor are there any applicable pharmacogenetics. The question of whether it's been interesting really is one that's entirely up to you. It's a question of taste. But I think that it's relatively interesting is that the genetic basis for human obesity, for people who thought about obesity 20 years ago, they might have thought about, if they thought about it at all, they may have thought about it as a metabolic disorder. And what it seems to be now is predominantly actually a neurobehavioral disorder. So this genetic work has kind of changed the way that we look at it. I'm not saying that there are not genetic variants influencing energy expenditure, and there will be, but in this first tranche of genetic discovery, genetics influencing body food intake are very strong. I should emphasize that many of the ways in animal models, if you manipulate many of these genes in the brain, you affect both food intake and energy expenditure. So there's a double whammy associated with many of these genes. So I'm going to move and switch gears now to go from above the neck, if you like, the causation of why we vary in our body mass and body fat stores between people, to why we vary in our susceptibility to becoming sick when we become fat. And the main reason we become sick is the very tight association with insulin resistance and the tight association of insulin resistance with other disease phenotypes, particularly diabetes. And it was thought for a long time if we could understand insulin signaling, we might really get our handle on insulin resistance. Now, human genetics has contributed to this. I've been involved in this game rather a lot longer than I've been involved in obesity. And the contribution of genetics to understanding human insulin action has been rather modest to date. Most of what I put on the slide here is derived from fundamental studies in cells and some in animals. And essentially what we understand, if we broadly looking here at how insulin controls glucose metabolism, I think it's very important when we define our terms. If you're talking about insulin resistance, you're generally measuring a glucose-related phenomenon, insulin's ability to lower glucose production from the liver or dispose glucose peripherally. There are many other actions that insulin has, which Robert Semple talked about this at his beautiful lecture yesterday. And here I'm really referring to insulin controlling glucose homeostasis. And it does so largely through a phosphatidylinositol 3 kinase-based mechanism, through a key core kinase called AKT2, which is upstream of many of the classical events of insulin action, and AKT2 being brought to the membrane by phosphorylated PIP3, phosphatidylinositol 3, and the phosphorylated in the third position, which brings AKT2 to the membrane when it is subsequently phosphorylated, and then it coordinates all the glucose responses to insulin. In terms of human genetics, human loss-of-function mutations have been found for more than 25 years in the insulin receptor, and a whole genotype-phenotype correlation of severity of phenotype with severity of mutation has been established, and we've been involved in that. There's a couple of disorders that were uniquely found by our group over the years. One family with a mutation in AKT2 leading to severe insulin resistance, another family with mutations in a protein phosphatase upstream of GSK3. I just draw your attention to a rare family we found a couple of years ago because it's become more interesting and more broad impact recently. This is an interesting couple of kids who presented with acanthosis nigricans of unknown etiology and normal fasting insulin. But a referring physician was smart enough to do an oral glucose tolerance test. And if you do that in a person with severe insulin resistance due to a receptor mutation, this is the pattern you usually expect: a very high fasting insulin, extremely high postprandial. What we got in these two kids was absolutely normal, in fact, even a subnormal fasting insulin, and massive postprandial insulin resistance, highly selective insulin resistance, not present in the fasting state, only present in the postprandial state. And that was because what we found was a mutation in this molecule AS160 or TBC1D4, which is involved in the trafficking of GLUT4 vesicles to the plasma membrane. It's what's called a RAB gap. And in the absence of a full moiety of AS160 or TBC1D4, you deplete the GLUT4 vesicles in skeletal muscle. So what you have is a human being whose liver works perfectly well, whose insulin works perfectly well, but whose skeletal muscle is unable to translocate GLUT4 in response to insulin, therefore produces an entirely postprandial syndrome of insulin resistance. We could find no more of these around, but it turns out that if you happen to be a Greenland Inuit, what used to be called Eskimo, then 17% of you have got a truncation mutation in this. And if you have it, and you have 2 copies of it, you have a 10-fold increased risk of diabetes. It's the first, they say, diabetes gene, which causes diabetes solely by impairing insulin action in skeletal muscle, discovered by Torben Hansen and published in Nature earlier this year. These people spend most of their fasting lives in severe postprandial hyperinsulinemia and have massive hyperinsulinemia, but actually there's no difference in their body weight, no differences in their circulating lipids. So it's a very interesting model for testing the hypothesis that insulin is a driver of bad things, because these people have a lot of insulin around for a long period of time, and yet other than postprandial hyperglycemia, they have very few other adverse consequences of their hyperinsulinemia. Whole exome sequencing in collaboration with Inez Barroso, again at the Sanger, and this time led by Robert. He will have told you about this syndrome yesterday. I'll just briefly touch on it, not to be repetitive. But I think this is just interesting to show you a sort of case. And a lot of the audience here are clinicians. Here's a young girl of Nigerian origin. Her mum's tall, her father's even taller. She's fully grown, but she's short and slim. But she presents with PCOS, a testosterone of 13.7, and mid-male levels of testosterone, very bad PCOS, and fasting insulin's over 20-fold elevated, severe insulin resistance. But unlike so-called typical syndrome X, she has no dyslipidemia, no hepatic steatosis on magnetic resonance spectroscopy of her liver. And when we give her stable water, we measure de novo lipogenesis. She's not producing any excessive lipids. This will be a very atypical picture for a severe insulin resistance of the syndrome X variety. And what she has is a truncation mutation in the P85 alpha subunit of PI3 kinase. It's now been found in many patients around the world with similar syndromes and shown to impair signaling in her cells, and also in an animal model that we've just created of a dominant negative insulin receptor. This is undoubtedly causative. And So just to step back from these defects of insulin signaling, I think one of the important lessons we've learned from them is that defects in how insulin works and how insulin signals does not actually fully phenocopy the metabolic syndrome. In typical metabolic syndrome, you not only have increased insulin, increased diabetes risk, but you almost always have high triglycerides, low HDL, low adiponectin, high amounts of liver fat. In all of these conditions, including the other ones I mentioned, the AS160, yes, you have increased plasma insulin, yes, you have increased diabetes risk, but all of these other markers of classical syndrome X are absent. And I would conclude from that it's very unlikely that defective insulin signaling per se is at the heart of what we think of as typical metabolic syndrome. So that means we have to return to ask the question, what is it that makes you insulin resistant when you become obese? It's not intrinsically obvious why carrying around 20 kilos of extra adipocytes results in insulin resistance, because we are talking about insulin resistance in liver and in muscle. And we have to somehow to translate that excess adipose tissue into insulin action in liver and in muscle. Because fat cells per se, if I give any of you 75 grams of glucose, maybe a gram or less would end up in adipose tissue. It's almost irrelevant for glucose clearance. So we have to understand that linkage. And if you asked 100 endocrinologists or 100 diabetologists these days, what's the common model that we think about as to what links overeating and to insulin resistance, they would probably tell you that the bad guy is this big bad fat cell and bad inflamed adipose tissue, because adipose tissue now is found to produce lots of adipocytokines, and one or more or a mix of these is the bad guy, somehow paracrinely or endocrinely influencing insulin action on liver and muscle. And this is, it's not quite a consensus, but it certainly is the common model. And I just wonder whether we're missing a trick with an older thoughts about insulin resistance. And that's one where our big fat cell is not actually our enemy. Our fat cell is our friend, because really it is the safest place in which to store excess energy. The real enemy is sustained positive energy balance, when we go day to day bringing in more energy than we expend. And our fat cell helps us buffer that until such time as we begin to reach a limit of our safe fat cell storage capacity, which may vary between individuals. And once we start reaching that fat cell storage capacity, we then start redirecting nutrients into tissues that should not have them, such as liver and muscle and pancreatic beta cell, and it's that's causing the severe insulin resistance. I suppose part of the reason I'm influenced in thinking that way is that we run a national service in England for the treatment and evaluation of patients who have disorders of fat, so-called lipodystrophy. These individuals have a developmental disorder or a functional disorder of adipose tissue. They're not thin, they're simply unable to make sufficient functional fat cells. And they really present a complete model of syndrome X. They have an accelerated full picture, including fatty liver, cirrhosis, hepatoma, including atherosclerosis, hypertension, early vascular death. Everything that you would want in a syndrome X is present in an accelerated form in patients with lipodystrophy. Now, in collaboration with my, again, like Robert X-Fellow and Robert and Sadat X-Fellow, and now PI and leading scientist in the Institute, David Savage, we've been involved in the discovery of 9, I think, discovery or co-discovery of 9 of the 13 genes that cause so far human lipodystrophy. But many of them affect many systems, and there are just 2 that I'd like to spend time today briefly talking about as exemplars of what you can learn from studying rare things. In fact, one of these syndromes is so rare that there's only one person in the world with it, but nevertheless, I believe it's very illuminating in terms of what it's teaching us. So I'm going to tell you a little bit about 2 of these genes that cause lipodystrophy. And the reason I'm selecting them is that the genes affected here only are expressed in adipose tissue. They do not function or are not expressed in any other place. They work, in one case, side C, only in white adipose tissue, not even in brown adipose tissue. So they're unique to the adipocyte. And what they're really unique to is the thing that is unique about the adipocyte, which is the large unilocular fat droplet, this amazing fact that 99% of the fat cell volume is taken up with a big wodge of triglyceride. It used to be thought of as the most boring cell on earth, given this homogeneous appearance of its huge triglyceride droplet. But of course, far from being boring, it's a beautifully regulated organelle within the adipocyte. And what it's there for is really to coordinate. This is the lipid. These are the two enzymes crucial for breaking down the lipid. ATGL breaks down triglyceride, HSL breaks down diglyceride, and then they release it when you're fasting, and when you need, they're releasing your energy as fat. They, of course, require coordination in their access to the lipid droplet, and perilipin and its partner molecule CGI58 are key to that. Perilipin, when phosphorylated by starving-related stimuli, brings HSL to the surface of the droplet. It releases CGI58, which goes to the surface of the membrane and pulls ATGL in a beautiful coordinated way. These molecules coordinate the response to fasting. There's another cell surface molecule which is not involved in lipase regulation, but is nonetheless essential for the formation of this big unilocular fat depot, and that's this protein side C present on the surface of the lipid droplet. And when it's not present, you can't make the big droplets. You make tiny droplets, as described by Mike Check and others several years ago. So you won't be surprised to hear we found human disorders of both of these, and I'd like to just tell you a little bit about what the human disorders look like. We now have many patients who have dysregulated lipolysis at the surface of the unilocular droplet. And here's an example of three relatives from a French family with three girls with very masculine fat distribution, dominantly inherited acanthosis, lipodystrophy, dyslipidemia, hypertension. And they have truncation mutations causing a truncated, partially functional copy of perilipin. And I don't have time to go into the biochemistry of this, but essentially what you've got is a continuous lipolysis, a continuous breakdown of this triglyceride droplet in an unregulated manner. Even in the fed state, this lipid droplet is continuing to release its free fatty acid into the circulation and causing, resulting in this continuous and toxic release of free fatty acids. And the single girl I'm going to tell you about is a consanguineous girl from Spain who actually has a remarkable syndrome, because very few cases of severe insulin resistance present with diabetic ketoacidosis, but she did. And she did so because you can see how much free fatty acid and glycerol she's releasing. Vast amounts of continuous release, because she actually has a homozygous stop codon in this protein side C necessary for making big fat cell, stable fat cell droplets. When you overexpress it in cells, you can't increase droplet size. It just, the truncated form doesn't work. And even biopsies from her, you can see that at least half her adipocytes have got these failed to form the safe unilocular droplets. And interesting, so I'll just conclude by both of these syndromes. What are they really telling us? They're telling us that all we need to do for everything that a diabetologist would practice with and see in his type 2 clinic, everything can be caused by the surface of the lipid droplet not being regulated in a temporarily controlled manner in an appropriate fashion. If you're not able to do that, if you're not able to store fat as fat when you feed and release it only when you starve, then this is sufficient to result in everything that we see and call metabolic syndrome. This was challenged by our basic science and colleagues, because two knockout mice had said, well, losing side C is a really good thing. If you knock out side C in an animal, they do really well, and they're protected. They're lean. They're protected from obesity. So we thought, oh, no. And David Savage, I have to take my hat off to him. He was incredibly brave, because it's very difficult to go and do negative work of someone else's previous good work. But he did, and worked with Peng Li, one of the authors of the previous paper, took the side C knockout mice, crossed it with the OB mice. And yes, they were substantially less obese than the OB, but when clamped, they were severely insulin resistant. So this was missed in the brain by not studying the animals long enough. And indeed, fascinatingly, it actually by knocking the side C mutation into the OB, you could actually cure the inflammation. These animals' adipose tissue were free of macrophages, looking beautifully clean, and yet totally dissociated from the severe insulin resistance you got when you disrupted its lipid handling properties, providing a blow, I think, for the met-inflammation hypothesis. In my view, practically fatal, but I'm challenging in saying that. And the rarity that we're talking about here is actually emerging as a relatively common phenotype in the general population. If you take alleles that predict fasting insulin in the general population and you create an insulin resistance score, you can then go back to other populations and say, what do people with a high insulin resistance score look like? They actually they get increased type 2 diabetes, they get increased hypertension, they get increased myocardial infarction, and yet they have lower body mass indices. They actually are almost as if they're in the population there is a form fruste of lipodystrophy, which underpins the predisposition to insulin resistance in the population. And this sort of lack of ability to store safely fat in adipose tissue may underpin whole, and I have no evidence to prove this yet, but the whole big questions like the susceptibility of South Asians to the development of insulin resistance at low levels of BMI may all relate to their limited fat cell safe storage capacity. So at the end, I'm going to go back to the diagram I showed you. How important are these various things? Well, genetics has told us that really our brain is a crucial organ for telling us how much energy we want to have in our bodies. And it does do so by influencing our appetite. It could do so by influencing our energy expenditure through sympathetic output. Surprisingly, our adipose tissue, which I say only clears about 5% of any carbohydrate load we take in, is yet disproportionately important in whole organism tissue homeostasis. Multiple pieces of evidence now pointing to the importance of healthy adipose tissue in keeping us healthy in the face of variations of energy intake and energy expenditure. Surprisingly lower impact than I think we might have seen in terms of genetic variation on molecules directly working on how insulin works in the skeletal muscle and liver, although that may reflect partly difficulties of ascertainment. And one thing I haven't touched on, because my colleagues Andrew Hattersley, Mark McCarthy, and others have really got a wonderful programs of work focused on human insulin secretion and islet cells, really important if you're not going to get diabetes to have good genes preserving your beta cell function, because most of the gene hits in the genome-wide studies for type 2 diabetes impact on pancreatic beta cell function. What about genetics? Isn't everything, because here is an organ that you haven't talked about at all, the enteroendocrine cells of the gut. If you're a practicing diabetologist, you'll be using therapies that manipulate enteroendocrine products of the gut more than almost anything else you've done differently in the last 20 years. And yet there's almost no genetic variation in those that may be so complex in genes like proglucagon with so many different effects that human genetics has certainly been silent on the impact of the enteroendocrine system. So just because some genetics isn't telling us something doesn't mean that therapy can't be important, and we should not forget pharmacology. But I would say that increasingly I'm becoming a little bit suspicious that as far as metinflammation is concerned, that the emperor may have, if not no clothes, be rather scantily dressed. Because so far neither rare genetic disorders nor common genetic variants, nor indeed much convincing pharmacology in humans, although a vast amount of work in rodents, attests to the fact that variation within the inflammation immune host defense system plays much of a role in either determining how fat or how thin we are, or how diabetic we become, or how insulin resistant we become when we do. And I'm happy to discuss that later with people. It certainly causes a lot of certainly attention when I've said it elsewhere. So I'm going to finish by basically saying that traditionally, and certainly growing up as a medical student in Ireland, our clinical geneticists were in a porta cabin at the back of the hospital, and they weren't given prescription pads, and they generally were looked in dysmorphology textbooks, and they gave names to syndromes, and we didn't really think of them as mainstream in clinical medicine. And the transformation over the last 25, 30 years has really been that genetics has come profoundly into practice in everything we're interested in, whether we're interested in height or calcium or thyroid or genetics. Extreme ends of human traits are likely to reveal previously unrecognized monogenic disorders. Some of these may be amenable to treatment. All are likely to illuminate physiological pathways relevant to man, and subtle variation in those are important contributors to more common forms of diabetes and obesity. I'm going to leave you with a quotation from William Harvey. Just a week before he died, he wrote a letter to a Dutch physician exhorting him to study the usual laws of nature by investigating rarer forms of disease, because we hardly perceive of things' utility until we're deprived of them. I'll finish by thanking many, many colleagues and collaborators over many years. I've been extremely fortunate, both in Cambridge and further afield, and it would be invidious to mention everyone who's contributed in the last 30 years since I've been in the UK. But I was very fortunate to have a wonderful mentor in Robert Turner in Oxford, who died far too young, and these things are often an occasion to remember the importance of inspiring mentors in in starting and catalyzing any career. My collaborators in Cambridge, and I particularly mention Chris Chatterjee, one of my longest-standing colleagues, and my ex-fellows and now PI colleagues, Giles Yeo, Tony Cull, David Savage, Robert Semple, who you heard from yesterday, and I'm sure you all know very well the stellar Sadaf Faruqi. I've been really fortunate in having fantastic trainees who have stayed around and helped make our institute the exciting place that it is. I thank my funders, but of course, above all, the patients and families who have contributed to the work that we've done over the last 20 years or more, without whom there would not be a program of research. So thank you for your attention.